Good afternoon, everybody. Uh, there are two speakers in the afternoon session. First is Dr. Malu, and second is Dr. Rakesh Srivastava. Now, uh, the first speaker will be Dr. Malu. I am introducing him. Dr. Malu is a learned teacher. All the times, he was Dean Rajasthan College of Agriculture, Director Research, Associate Director Seeds and Farms, Director Research. He, Dr. Malu has developed many new high-yielding crop varieties of durum wheat, small millets, groundnuts, and seed species. He handled a number of research projects of national repute as a principal investigator and headed the Department of Plant Breeding and Genetics and Molecular Biology and Biotechnology for more than 6 years. Thereafter, he joined as a Dean and Chairman, Faculty of Agriculture Science, Pacific University, Udaipur. He is also an adjunct professor of RSQVB, Gwalior, member of BOM and selection committees, MPUT Udaipur, nominated by the government of Rajasthan, Jaipur, and he has a vast research and teaching experience in the university and in the state. And he also handled the number of research projects in the university, that is of ICR, DBT, RKVI, DST, and international projects also. I request him to deliver lectures. Please carry on, sir.
Okay. Esteemed Chairman, Dr. Khandelkar, Honorable Vice Chancellor, is it audible? Okay. Honorable Vice Chancellor, sir, Dr. Rao Saab, distinguished speakers, I'm sorry, only Dr. Rakesh Srivastava is left, no? And Dr. Bharadwaj, Dr. Meeta, and Dr. N.K. Singh, all of them. Dr. Tripathi, Dr. Tiwari, and so many colleagues at Rajmata Vijay Raje Scindia University, and so many distinguished friends and students. Can I switch over to the slides, sir? Yeah? Because it has to be done right from there. May I have slides? So thank you very much for giving me this opportunity to be amongst you. And it's a webinar on the application of biotechnological tools in crop improvement. And here I'm going to highlight not anything new, but the kind of accomplishments which have been made in our system, in our country, the challenges and the strategies for the sustainable agriculture. Sir, our country, next one. Sir, our country is unique in the world in many respects. It has a great history and dynamic culture spanning back to the beginning of the Indian agriculture, beginning of the human civilization. Indian agriculture began by 9000 BC on the northwest India, as a result of early cultivation of plants and domestication of crops and animals. Can I have the next one? If you see, sir, our country has a lot of resources, a treasure of resources. About 11.3% of the world arable land is with us. 18% of the world population, of course, it's not something rare, but okay, it's having another advantages. 14.7% of the world cattle, they are with us. 4% of the different water resources of the world are with us. Four hotspots of biodiversity, and the country is having all climatic conditions. That's we are blessed with those things. Next one. Our country is a mega diversity center and accounts for about 7 to 8% of the recorded species of the world, spread over 45,500 species of plants in different habitats. Next one, coming to the agricultural revolutions which have taken place in our country, I should say in each and every sector of the agriculture, we have witnessed the revolutions. It can be but the major breakthrough we have obtained in the green, white, yellow, and the blue revolutions. And India is the second largest, you can say, the agrarian economy of the world. And the food grain production, if you see it right from 1950 to 2020, that has increased from 50 million to 300 million tons. That is 6 times. The, that has been, earlier we were having shortage of the food, now we are having surplus. Food grain includes everything where wheat, maize, barley, or the rice, pulses, oilseeds. In case of the horticultural crops, again, there is a 9 times increase which we have witnessed, and it has gone beyond 320 million tons. Milk production, we are number one in the world, and it has increased from 17 to 184 million tons. That is a big breakthrough of about 10 times what we were achieving in 1950s or 51. If you take the fish production that we call the blue revolution, it is again 14 times, 0.75 to 12.6 million tons. The poverty part, if you see, the reduction has taken place from 70% to the 20%. Export of food grains, that has increased up to 20 million tons. And this time we are going to witness a record production of the food grains. So that's a big achievement. Next one. Here, right. If you see the position of the country in the world, we are number one in the arable land. Just I highlighted that we are number one in the milk production. Population, we are two, next to China. Fruits, again, second. In case of the groundnut in shell, second. Sugarcane, we are second. Vegetable and melons, again, second. Cattle population, we are second. Cotton, again, second. And total cereals, we are third. If you see, next one, the growth trends of the last 15 years, you can see that in the first phase from 1970 to 1960, 86 or 85, 86, in 15 years, the cumulative agricultural growth rate was 2.5%. Later on, in the second phase, it was 2.79%. And in the last 15 years, that is from 2000 to 2015, it has 3.97%. But looking to the pressures, it has to be increased beyond 4%. The increase has been just finding some of the achievements in terms of the green revolution. The big change we have because of the discovery of those dwarfing genes, the Pusa Basmati 1509, and similarly Pusa Basmati 1121. These varieties, they have shown outbreak in terms of the production, particularly 1121. It's a medium duration variety, and the yield levels have gone up to 5.7 tons per hectare. And single variety, particularly to this 1121, has generated revenue of about rupees 1.5 lakh crores just in 8 to 10 years, 2008 to 2016. Then similarly, similar to the rice in wheat varieties also, HD 3086 was the another one variety. HD 2967 was another variety of the wheat, which was the choice of the wheat farmers. And that has given a production up to 15 quintals per hectare. And it has shown its worth in the northwest plains, and it has given up to 44 quintals in the eastern plains. And this HD 2967 is the only variety which has been for the last 4 years having highest breeder seed production. Consequently, in the last 4 years, so miracle has taken place with the newly developed variety. Similar to the wheat, we are just as it is visible, India is a leader in horticulture also, and you can see the crops: banana, grapes, papaya, tapioca, lemons and lime, cabbage and other brassicas. Here we are leaders in the market in the entire globe. So things are going on in such a way that India has become a leader. If you see white revolution, for which country is known, Dr. Kurian is the mastermind for that, and you can see that he has given an example of modern value chain management. Dr. Varghese was awarded World Food Prize also, and about he has linked up about 15 million of the millions of the farmers, mostly called smallholders. And you see that chain, that milk production producer starts, then it goes to the village dairy cooperative, then it is the district milk cooperative union, then the state cooperative milk unit, and finally it goes to the consumer. So it's an exemplary, you can say, achievement which has been made by our country. Next one. If you see the use of the cotton, country used to be a cotton producer, great producer, but in the after 2000, there was a big change in the scenario of cotton production, and a big revolution has taken place. Of course, hybrids have been developed by the country, particularly the C.T. Patel is known plant breeder, known as the father of the hybrid cotton. And afterwards, the BT come, and then about now 92% of the total cotton is under the BT hybrids, and they are being grown over 11 million hectares. Similarly, 7 millions of the 8 millions farmers opted BT cotton. Mostly they are the small holders. So sevenfold. The another interesting part is how we saved our environment, because cotton is very susceptible to insect pest diseases, and sevenfold reduction in the pesticide use has been observed. 30, you can say 9 to 10 million bales exported each year, and valued of the, about $3 billion. And Cotton has generated millions of additional jobs in the country. Next one. If we come now, enigma. The Indian enigma is that now we have seen so many revolutions, but despite of those revolutions, a rapid economic growth trajectory of 7 to 8%, one fourth of the world's hungry and poor population, or you can say 200 million, they are the home of the India. 40% of the world malnourished children are our own. High epigenetic and economic cost. 9% of the total GDP is being lost annually. One fourth of the world's smallholder farmers are in India, and they comprise 50% of rural population, poor or hungry, and particularly the women farmers are mostly deprived. It is we are known for the nation of youth, but largest bulge of unemployed, poorly employed youths are there in terms of the demographic disaster. Agriculture three times more effective than other sectors in alleviating agrarian crisis. Yet the investment in agriculture is very low, you can say. It not only preventing malnutrition, that delivers about 16 to 40 million dollars on return of investment for every $1 spent. So, which we say that the pot of gold, justice, inclusiveness, balanced nutrition at the end of the rainbow remains illegible. Next one. The reason is you can say yield of the major crops. If you see the data in the countries, if you compare it with the US or the Korea or Japan, China, European Union or the Brazil, here in India, wheat, we are just, you can say, 30 quintals. Where the other countries, if you see, even the Korea or the Japan, they are better than us. Smaller countries. We are, in case of rice, we are just 25 quintals. I think we are at the last out of these countries. In case of the maize, it is 27 quintals. And if you see it with the US, we are just almost even not half of that. In soybean, again, of course, Madhya Pradesh is a state which is a bowl of the soybean. But again, here the productivity levels are just 9 quintals per hectare. So there are gaps, and particularly the gap is particularly at the vegetable oil economy. Current annual requirement is 22 million tons, and what we are importing annually, that is 70% of the required, that is 15 million tons. Cost of annual import is rupees 75,000 crores, and the projected demand by 2050 will be 39 million tons. So you can see there is a big gap. If you just compare the average yield of the other oil crops, soybean we have seen, but if you compare it with the other the world, soybean is just half of the world average. Groundnut 1.24. In case of this mustard 1.25 against 2. Sesame we are almost 3 times less than the world average. Safflower again 3 times less. Sunflower again almost 3 times less than the world average. So there is a big problem with the oilseed crops. Next one. So small farmers, because we have small holdings, so you can see the difference. How many farmers are putting their effort in generating, in what way they are producing. So the simple picture shows that we are under the stage of hungry, and that is a biggest that we have to uplift our marginal or the small farmers. Next one, per capita water storage, because in agriculture, if you see, water is very important, and without water we cannot have anything. But we are not harvesting water. That is the important thing which we want to highlight. We are our total crop production, food grain production, all every sector of the agriculture is affected by the water, and still we are dependent much more on the monsoon. We are not having storage capacities. So you can see in comparison to Russia, Australia, Brazil, US, China, South Africa, we are at the minimum. India, where the per capita water storage capacity cubic meter is extremely low. Next one, if you compare the progressive expansion in the occurrence of nutrient deficiency, the soils are getting depleted in terms of the macronutrients, in terms of the micronutrients. The data, they are self-sufficient to highlight that coming now the soils are almost starved. In terms of we are extracting, absorbing, and each and everything, nitrogen, iron, phosphorus, zinc, potassium, sulfur, manganese, boron, molybdenum, copper, every time another element gets up, adds up. So the soil condition is also, you can say, it's alarming. So we have now, next one, we have so many future challenges. The population is of course one, and which is going to increase, which is increasing at an alarming stage from 1.7 billion. It will be around at 2050, and we will be the most populous country. We may leave China behind. So there is a need to increase the food production by 70%. We have to 70%. We have to increase from that limited area, that is the land, water, and labor productivity has to be increased by land in terms of 4 times, water 3 times, and labor's productivity 2 times. So population is unethically highly undernourished. There is a big gap, wide inequality between the rich and the poor. We are much more affected by the climate change. Perhaps we are witnessing the first part, and the production resources are shrinking day by day, and we want to have more from less for more. So there are so many challenges, and the crop management challenges are that may include so many things. The water management is the most one important one. Less subsoil water is there. Besides that, soil health, declining water table, or the increase in the incidence of diseases, insect pests, reduction in the farm profitability. We are exposed to, in the just last 2 months only, we are exposed to Ativristi and Anavristi. You can see Amphan, you can see Nisarg, super cyclones, storms, locust is there now. Even MP and every part of the country is facing the locust, tiddy, that is there, and beyond all the COVID-19. So that has become the major challenge. Labor is very costly, and we can imagine the fate of our economy as most of the labor they return to their home. So here we have to can we think in terms of the farm machinery, storage of vegetables or the fruits, or their transportation. They are all challenges. More cold storage are needed. Then there should be the cold trains, the trains with the refrigeration. And the biggest problem is the farmer is being cheated by the middleman. So they are making money. So farm gate to the finally A to Z, that is the mandi. That's the dire need in the present scenario. Then if you see, I was just talking about that climate change, no? And if you see the projected changes in agriculture productivity from this, you can see we are tried to compare with the other countries, and India is mostly vulnerable at the vulnerable stage. You can see Australia or Canada, United States, China. Even China is not being much affected, but we are going to be having the great impact of climate change. The other things are there. And if you see the United Nations Sustainable Development Goals aim to leave no one behind and directed all the countries of the world to work together in the team form to end the poverty and other deprivations. And India is, of course, committed to meet sustainable development goals. And in terms of we have to increase the farmers' income, we have to think of the diversified and the sustainable agriculture. So now the new vision for agriculture, or we can see what should be done. So how to meet the sustainable development goals by 2030. Productivity has to be enhanced by 70%. We have to reduce the poverty by 100%, hunger by 100%, emission of various greenhouse gases that has to be reduced by 35%. And for this, many policy decisions have been taken up at the level of government of India, at the level of state governments to increase the farmers' income. So many new initiatives have been taken. In that, ICR has also taken up, given a big farmer first program, FFP, farm innovations, resources, science and technology, initiated to enrich the farmers beyond the production and productivity. Besides that, it aims to integrate the technology. So we have to, and the efforts have been made to harness, enhance farmer scientist interface, and how the science and technology can be effectively harnessed. So the kind of strategy which has now on which the work is to be taken, we have to revisit our government policies or the reforms for the secure, sustainable agriculture, and accordingly, investments or the infrastructure be made addressing the two most important things: climate change and the soil health. And of course, the water is there. So we have to review our national agriculture policy, which was chalked out long back in 2000, that is about 20 years back, and the national policy for the farmers in 2007. So there is a need to relook it. We have to focus on a very big section of the farmers, and that is the small farmers, which have the small land holdings. Diversification is the dire need. Farm production to value addition, that has to be done. Then market reforms have to be improved, and the linking farmers to the market. Recently, just about a week back or 3 days back, an ordinance has been passed by the central government. One nation, one market. Now farmer can sell his produce anywhere in the country directly without the middleman. That's a great achievement that has been made, and that has been done with the help of the Niti Aayog. The government of India has taken decision. Then e-mandis or e-nam is there. Now they have depleted or removed the mandi tax also. So there is an now uh, freedom has been given to the farmers to sell it, produce. Even they can send it to any other country of the world. So effective coordination, of course, we have so many schemes, but there is a need to have the effective coordination. There are so many projects are there. The need is there to have the linking up those things, projects, and regulatory bodies. So we have to work in the mission mode approach for strengthening the precision agriculture, because now you have to be serious about artificial intelligence. You have to think of drones. Now the TDs or the locust is there, and we have to, and efforts have been made that now drones are being utilized to spray insecticide. So many other new implements are required. Farm machine mechanization is the need. In fact, the agriculture is based over the six pillars, and I think all those six pillars, right from the soil or water or the mechanization or the implements, then the climate and the farmer, they have to be individually improvised. Livestock is there. We have the fisheries section. There is a need to strengthen and work over these different aspects of the secondary agriculture, or you can say so many other things. Different names have been given. The CA has emerged of the conservation agriculture. So these fields are to be strengthened. Quality seed is still a thumb or the most important out of these critical inputs or the planting material. And effective use of solar power, we have plenty of, our country is blessed with that lot of solar, and we have to utilize it. If you see the just productivity difference in the precision and the non-precision farming system, there is a big gap. Next one. If you see particularly the next one, the if you see working the new system, particularly the mechanization has come, and here happy see that has come. The straw burning problem has reduced now. No, please back. And high clearance that with the help of the Punjab Agriculture University and the Borlaug Institute of South Asian Studies and the private sector, the seeder has been developed, and we have saved a lot of energy and manpower, labor power. Seed, you have seen the country's seed man among the Dr. Rao is sitting, the man of he's a seed man. You can see a revolution has been done, and still with working with two three projects, it has been the power of seed has been highlighted, shown that has tremendous effect on the economy and the agricultural production. So quality planting material has a has done a great service to to the mankind, particularly the Indian community, and we have to further sensitize the farmers. We have to make the farmers seed producing companies. We have to give a lot of training and capacity building programs. But similarly, our seed act has to be now, at least it is pending. It has to come. If you see the solar power, that's wonderful, and we are blessed with that one, which I told you. And now we have to harness the solar power in terms of making agriculture as a boom. So science-based agriculture is now required for commercial agriculture. That is the we have to think in terms of the commercial approach. We have to think in terms of the GM crops. Even Dr. M.K. Singh was telling that the Bangladesh has been given to grow the commercially they can grow the beauty brinjal, but in our country we cannot, except cotton. Everything is in testing stage or some or other hurdles are coming up. The Department of Forest and that those so many other NGOs and other things are coming up. Molecular farming is the need of the hour, and the university, particularly this Gwalior University, is putting a lot of efforts in terms of at least bringing all the scientists working on the molecular farming or the biotechnological tools. They are together today with us. Bioinformatics is required. Now the technologies like CRISPR-Cas9, nanotechnologies, they are to be exploited. We have to explore plant microbes. Now it's the time when their requirement, they can improve so many things, including the soil or the seed or its germination, or so many things are there. So it's the time of the microbes in terms of the insecticides, pesticides, improvising their efficiency. Then we have to strengthen some of our existing institutions. Particularly, we in the world we are having the biggest national agriculture research system, but it has to be strengthened in terms of we are having more than 102 ICR institutes, more than 75 SAUs are there, and so many villages and the scientists and everything is there. So we have to furnish, equip them, and looking to the present situation of COVID and looking to the food condition, because it was our agriculture which has saved us this time. We were having everybody was running for the lockdown during lockdown period. Everybody was of the opinion that let me have the safe food for me, or the vegetables or the fruits. So now people have realized that it has to be strengthened. We have to strengthen our panchayati raj systems. We have to the government has to give more responsibilities and authority for the agriculture development to these institutions, particularly panchayati raj. Extension agencies, particularly I am talking of the state government agencies, they have to be operationalized. Atma is there, Agriculture Technology Management Agency. So now everything they have to be put together. So there is a need to scale up the things like nutritional security using science technology interventions on biofortified crops. The QPN or the hybrid technology has given us the QPN where the tryptophan lysine content has been increased. Dr. Wassel was given the World Food Prize for that. Now, in case of rich rice has been developed, having which is fortified with the zinc and the iron. Zinc wheat has been developed. Some of the varieties have been developed, of course, but there is a need to further strengthen those research aspects. Dr. Tara will be talking about the iron-rich bajra, and there is a need, and we have to further strengthen these. We have to fortify our food. There is a need to develop region-wise, crop-wise, integrated crop management strategies for holistic approach to sustainable agriculture. Now, involving all sectors, including agriculture departments, or the universities, or district administrations, or the industry people, and the farmer. So, a team has to work, and it can be even at the district level, or it can be even at the tehsil level. So, we have to strengthen our integrated crop management strategies. using diversification also. Then we have to bridge the gap between the farmers and the technology developer. Of course, the programs have been launched lab to farm to lab or lab to farm. Everything is there, but still there is a big gap, and it has to be narrowed down. Then you have to relook a relook the role of the KVTs. We have to start agri clinics. We have to increase or we have to initiate mobile advisory units, just what Amul is doing. Farmers meeting, it should be strengthened. Field visits be the farmers are to be given so many field visits. They can be sent from one end to the other end of the country. There should be some farmer science congress also, and that can help us to integrate the traditional and the modern knowledge. We have to think in terms of the farmers producer organizations. We have to sensitize the kvks in terms of for involving them in the planning and research and extension activities, their capacity besides their capacity and hands-on training activities. You have to think in terms of improving the I should say the improving the skill powers of the tribal population. There is another scheme which has been taken up, NARI, that is the Nutri Sensitive Agri Resources Innovations, or the Vatika Value Addition and Technological Incubation Centers in the agriculture, funded by the RKBY. So, like this, we have to give. Now, so finally, our health. You can say, no, just back, please. So, one health is there. So better health, better soils, better plants, then better animals, and the better human beings. So you can see all the things they have been put up in case of plants: biofortification of the genotypes, crop selection, crop management, grain and feed quality, which I tried to summarize it here. In case of animals, healthy animals feed on the quality fodder or the forage. We should increase the feed mixtures. In case of the humans, balanced, diversified, and healthy diets. So all that's we have to think in terms of those things. Then the Sansad model, next one, of the developing climate smart villages, that is local to global. That there are climate information services there, local adoption plans should be there, climate smart technology should be there, index based insurance, and they can be the learning sites, multiple partners, you can say capacity building, and finally it will scale up policies, private sector, extension mainstream success via major initiative. So that is being nowadays it is called the Sansad model. And finally, for all, we need a wonderful entrepreneur, farm entrepreneur. And a farm entrepreneur should have so many qualities, like it should have the confidence, flexibility. The core values includes trustworthy or honest, problem solving, derive, drive in terms of the highly motivated, highly energized. He should be determined or persevere. There should be a competition, that is the competitiveness. He should take initiative, goal driven. And in this respect, so many schemes have been floated. Even ICR floated a scheme, ARIA, attracting and retaining youth in agriculture. Here, the role of women has to be centralized. We should empower women for making them an entrepreneur, particularly farm entrepreneur. And here we have to think in terms of how we can develop motivate the youth, particularly in the farm sector. And it is a time after COVID-19 that our all laborers or the workers, they have reached their home. Now they have to think in terms of agriculture, where they can think in terms of so many other different units of secondary agriculture, putting them, which they can become the entrepreneur, and we can strengthen the economy of our country. Next one. So, summing up, the only most important thing which come to my mind is we are very good in concepts and planning, but not so efficient and effective in execution as the agriculture matters the most in India. So, therefore, interdisciplinary and integrative approaches, adequate investment, vibrant science technology interventions, so many innovations, trained human resource, responsive and effective implementation are, you can say, they are must to reach the unreached so as to build India as a $5 trillion economy. And our father of the nation, we must be guided by following his profound words. Mahatma Gandhi highlighted that nation judiciously be a blend of knowledge with the character, commerce with the morality, science with humanity, and policies with the principles. Politics, particularly, not the policy, politics with the principles. So, thank you very much. And I'm really grateful to Honorable Vice Chancellor, Dr. Tripathi, Dr. Tiwari, for giving me this opportunity to be a part of this national webinar. Thank you very much. Take care.
Thank you, sir. Most vast, informative, and policy driving lectures to our students and delegates. There are some from the delegates. Please clarify it, sir.
Uh, right now, I'm getting some questions. Can I respond to them, sir?
Yes.
Okay. The question is how forestry can be used. Of course, forestry, we consider forestry as a part of our agriculture. Of course, there are universities, separate departments are there, but forestry and the agriculture will go side by side, not only for the tribals, but it is a part of our livelihood. So, governments, there are so many government schemes, and particularly but in different sectors in different states, the schemes are there, and they can be helpful in terms of the, in case of Rajasthan or MP, there are tribal communities are there, and the forestry is well linked with the agriculture. Special schemes have been launched. There are incentives have been given, and they are in all PSPs, and they are all being funded by the RKVY, Government of India. So they are there, and that is the main part, and they will go side by side always.
Thank you very much, sir. Have a seat, sir. Namaskar. Now, the next speaker is Dr. Rakesh K. Srivastava, Principal Scientist. He will deliver a lecture on per millet genomics and molecular breeding. Now, I am introducing him.
There's a lot of ego. Maybe somebody has to mute
He is
his or her
a
mic.
Principal Scientist, Genomics and Trait Discovery Research Program, Genetic Gain, ACRISAT, Hyderabad. He led the per millet genomics and molecular breeding team at Ecrisat. He is extensively involved in the generation of the cutting edge genetic and genomic resources, mining, mapping, and deployment of QTLs genes for various biotic and abiotic grain and fodder quality related traits in pear millet as a global mandate. He led a team comprising of national and international partners to define the heterotic gene pool in pear millet for the first time. He also led the pear millet downy mildew genome sequence. He also contributed the word reference germplasm tip 23D to B1 PIP5 for deep sequencing, which now serve as a word reference genome for permalate. He worked with the Syngenta seeds for 2 years after his PhD before joining the Agrisat in the year 2006. He is with the Agrisat for the past 14 years. Is currently working as a principal scientist. He has more than 110 publications, including journals like Nature, Biotechnology, Scientific Reports, and many more. Thank you, sir. Please deliver your lecture.
Hello, everybody. Can you all hear me loud and clear?
इनका स्क्रीन
इनका स्क्रीन लाना है इनका राकेश श्रीवास्तव सर
Screen sharing मैंने कुछ नहीं किया है. So if you can just be allowed to speak for some time before I start my presentation.
डॉक्टर राकेश
इनकी आवाज नहीं आ रही है
ये म्यूट है.
Hello.
राधेश्याम शर्मा
Mute नहीं है मेरा. My mic is.
नहीं ये मिटाना है.
आएगी ना, आवाज आएगी. एक मिनट सर प्लीज.
प्लीज वेट सर.
आवाज इसलिए नहीं आ रही क्योंकि अपने माइक सारे स्पीकर्स म्यूट है. अच्छा अच्छा. अब वो बोल रहे हैं. एक मिनट तो आप म्यूट कर. हाँ प्लीज सर बोलिए.
Okay. Yeah. Thanks very much.
राकेश welcome.
Yes, please. Thank you very much for this wonderful opportunity. This is last presentation of the day, so I'm not sure, how many of you are still online, but I would try to make this presentation as interesting as possible. So let me begin with thanking the Honorable Vice Chancellor, Dr. S.K. Rao Sahab, who has been our long-time collaborator and also our well-wisher and friend. It's really very nice to see you, sir, after a long time in good health. And thanks very much for this wonderful opportunity. I also thank the chairperson of this particular session, Dr. Khandelkar, and my previous speakers, Dr. N. Singh, who is also my teacher. In fact, when I was doing my PhD with Dr. Mohapatra, I used to go to his office, bug him pretty often about my doubts. And I was the only one not working on rice. I was working on peas in the labs of rice genome sequencing project. And I'm really very happy to see my classmate, Dr. Amita Mitra. So it's really wonderful to see great speakers, including Dr. C. Bharadwaj, who is just not a good friend of mine, but also he is my Guru Bhai. And lastly, it was really very nice to hear Dr. Malu. He covered so many things so well. So especially Dr. Bhardwaj, on my part, I was actually joking in the lunchtime that he hasn't left much for me in Pearl Millet section. He has already covered a lot of things. So he has really made my job very much easy. And, trust me, I was actually modifying my presentation, since morning, because many slides were kind of common slides. And I just thought maybe, after these presentations from Dr. N.K. Singh, especially, and also Dr. C. Bharadwaj, some of these will become redundant. So I tried removing them, but please bear with me in case you see something which is kind of duplication. So with this background, can I just go ahead and start sharing my screen, please?
Please, sir, share your screen, sir.
Yeah.
Okay, please share.
So my presentation is online. I mean, is it on the screen?
शेयर नहीं हुआ सर स्क्रीन शेयर पे क्लिक करके और फिर उसका शेयर बटन पे एक्टिवेट जब हो जाएगा ब्लू कलर में तब क्लिक कीजिए सर
एक मिनट Yeah, all that is done.
Okay.
Are you able
Okay,
to
now,
see?
sir.
Yes?
Yes.
Okay.
Yes.
All right. So here I go. Today, this is such a great pleasure and privilege to be standing before my teachers and before my mentors and also my friends to speak before you a crop which is kind of, shadowed by, mega crops like rice, wheat, and corn. So I think this is a good opportunity to know about the advances we have made in pearl millet. And I will take you through journey of my slides in the next 50 minutes or so, and we'll see what exactly we have done. But then what I did is, I actually made my presentations into two halves. One was actually related to development of genomic tools and technologies, which I thought would be important for some of the speakers. And other section was specific to pearl millet. So after seeing presentation from other speakers, I thought, I should cut down on that. So I tried cutting some of them, but please bear with me. But before I start, let me just let you all know what ICRISAT is. Some of you might not be knowing. I'm not sure how many people are online right now. So ICRISAT stands for International Crop Research Institute for the Semi-Arid Tropics. It's located in outskirts of Hyderabad. Of course, now city has expanded, so it has become very much part of Hyderabad. It is spread in in vast area, almost in 3,000 acres. It's a beautiful campus, one of the most beautiful campuses of the CG centers. And this is part of the consultative group on international agriculture research, an umbrella of 15 international organizations, which actually do research on various areas of agriculture. So we have our regional hubs, three regional hubs located in WCA, which is Western Central Africa, and 4 regional hubs in Eastern Southern Africa regions. And you can see actually ICRISAT works in all these, shaded areas, which are shown in the shades of red. And it covers about 55 countries and touches about 6.5 million square kilometers, and is an important, important vehicle in translating the upstream science to downstream, delivery, which Dr. S.K. Rao was talking in the morning. So ICRISAT actually believes in production of international public goods, which are knowledge, which are also cultivars, which go into farmers' field. So I think my previous speakers have given background why we need molecular technologies, but let me also touch base on this maybe from a different perspective. So as we all know, we are sitting on a population time bomb, which would explode at certain point of time if it is not actually controlled, especially from these two big countries, India and China. And every day we add about 200,000 individuals, which is like 2 lakhs. And so far, since midnight, we have already added about 140,000 people, about 1.4 lakh people. So it is definitely a worrisome situation, and we need to keep pace with the ever increasing population. And if you see effect of this rising population on the resources, the kind of pressure it puts on the resources of Earth, it is definitely an alarming situation. The kind of waste we generate, it is so much, it's enormous. And in some paper which I can quote separately, they have calculated that we need 1.7 Earths actually to take care of all this waste which we generate every day. And on a cosmic scale, if you see, the universe was born almost like 14 million years ago, and Earth was born about age of universe is 14 billion years ago, and the age of Earth is about 4.5 billion years. So if you were to contract 4.5 billion year, into a 24-hour time frame, then you would be appalled to see that out of the 23 last minutes where life started to exist, we have used 1/3 of Earth's natural resources in just last 0.2 seconds. So this is an alarming situation, and we need to get into practices which Dr. Malu actually presented very nicely, which lead to sustainable growth and development. So we need to find out ways and means to evade this Malthusian catastrophe, which is actually waiting, which might explode at any point of time if we don't take care of all these things. So we have these molecular technologies, which are part of the big picture, which also contribute in a big way actually to take care of this growing imbalance between rising human population and food supply. So now coming to plant breeding, Dr. N.K. Singh very beautifully mentioned about this. So now we see that there is a revolution in the way plant breeding actually is taking place, especially if you see the technologies which have been generated in last say 10 years. You would see we have actually taken leaps and bounds over what we are doing previously. So earlier breeders really did very good job, and we call it as eye of the breeder. Some breeders actually have that I would say intuition, or that experience or knowledge, which actually help them to select the required phenotype. So that is important. But at the same time, everyone cannot actually do that. We need to have support which actually comes from the gains in technologies we have made. So this is what molecular breeding is all about, and this is beautifully depicted by this iceberg picture which you are seeing here. So earlier we were targeting just tip of the iceberg, whereas the base, the 90% mass which actually floats below the water was totally untapped. And with the current technologies, wherein we actually work on machine learning, on artificial intelligence, and we make use of the big data which is actually generated, which basically comes from the phenomics and genomics platforms. We are able to find out the big chunk which actually floats down under, which are the small effect loci, additive loci. We actually go for whole genome scans, and we are able to find out the markers which are actually linked to regions which actually don't contribute in a big way. So we are able to actually get hold of all the chunk which was lying undetected previously. Now this is the breeder's equation, which I'm sure many of you would have seen. I think Dr. Bhardwaj presented this very nicely, so I'd not like to get into detail of this, but excuse me. But if you see here, so here there are certain terms on the numerator, and there is a value on the denominator. Now, on numerator, you see you have selection intensity, you have heritability or prediction accuracy, and you have the genetic variance. And on the denominator, you have the breeding cycle length. So, one would ideally ask where, as a plant breeder, I would like to invest my resources in. So I think we at ICRISAT have had a lot of discussion on this, and we realize that although the terms on the numerator are important, but the one which is on denominator is extremely important, because it is just one term, and if you are able to actually contract the length of the breeding cycle, then you can actually achieve enormous genetic gains. And in this direction, as Dr. Bhardwaj was mentioning, we have a world platform which actually deals with fast generation advancement. And this has been optimized in many of the ICRISAT crops, and it can actually be expanded to outside ICRISAT crops as well. And this is done by tweaking various parameters, growth parameters, which include things like wavelength of the light, moisture content, the fertility gradient, and things like that. So if any one of you actually are interested to develop this kind of platform at your end, you are most welcome to liaise with ICRISAT, and we would be in position to supply you all the technology which we have generated at our ends. So this is again that famous slide which which actually is from Dr. Rajiv Varshney's paper published in Current Opinion in Plant Biology, and shown by Dr. Bhardwaj. So here, I'm not going to add anything more. See, our area is starting from 1G to 4G. 5G is something which is maybe on other side of the road. So my presentation would not be talking on the 5G, although it's an exciting area. So as a breeder, we must have complete understanding of the germplasm, and see, nowadays there is a kind of I would say mad rush to sequence everything, which I think is not really a great decision, because see, obviously sequencing costs are coming down, but at the same time, you got to make use of your resources in proper way. So the first thing is to understand your germplasm and your genetic stocks. And once you have understanding of your germplasm and genetic stocks, and you want to get them sequenced, either whole genome resequence or maybe a reduced representation kind of sequencing, then maybe you will be able to understand the genome, and from genomes you go to the genes, and from genes you can actually go to genomic assisted breeding. Now, so this is again just to support what Dr. N.K. Singh was trying to tell in the morning. So we have gone a long way, and Dr. Bhardwaj also mentioned, from isozymes to these next gen markers which are SNP based. So currently, looking into the cost and throughput, and these SNP markers have become choice of markers. And the genotyping now is purely, sequencing centric. It's all automated. And as Amita was mentioning, the error rate is very, very low. So whatever fog or whatever noise actually you get to see, it actually comes from the phenomics platform, and we need to actually do a lot on the phenomics side. Now, coming here on this side of the road, we have these sequencing platforms can be actually divided into two major platforms. One is reduced representation that details are given. I don't want to get into all those. And second one is your brute force, wherein you do whole genome resequencing, and all these actually with cost. Depending upon the availability of resources at your end and your project goals, you should take a call which exact platform you should be using. And ICRISAT also has a genotyping unit led by Ms. Anu Chitkinini. She actually is part of the Genetic Gains program. I'm also part of the Genetic Gains program. So if any of you would like to get help on the genotyping side, you are most welcome to get in touch with us. And we also take care of DNA isolation. So as Dr. Bhardwaj was mentioning, you don't need to really get into all nitty-gritties of isolating DNA. What we need nowadays is just a leaf disk, a couple of leaf disks to get things done at our end. So I just thought maybe I should also touch base upon the rapid evolution of sequencing technologies. So as Professor Enkesing was mentioning in the morning, we have gone a long way, starting from first generation Sanger sequencing, where the cost was huge and also the turnaround time was kind of very large, to the current third generation or next generation sequencing platforms, which include platforms like PacBio and Nanopore. And these technologies actually help you produce long reads against, say, Illumina, which produces just about 100 to 200 bases, versus what you get here, like 13 to almost 20 kilobases long read. And the good thing is, the error rates also of these platforms are coming down drastically. So there is a revolution in the technology. So you can have the best of both worlds. You can actually combine your short reads with long reads and really can generate good quality genome at assembly level, or maybe you can just go ahead, if you have expertise, you can even generate a gold standard reference genome. I thought maybe I should also let you know about some of the options which are actually available currently, which relate to high throughput genotyping technologies. So this includes your Nextra i-through-seq, provided by Novogene, BGI. Then you have option of Nanopore, provided by Oxford Nanopore Technologies. You have DartSeq coming from Enrique Lian's lab based in Australia. Then you have RAMSeq. So all these have advantages and disadvantages, and based on your requirement, you should take a call as to which platform you would like to go for. So one could be used for discovery platform, other could be used for development of maybe whole genome prediction models, wherein you don't need kind of high density markers. So there, this RAMSeq I think holds a lot of promise. Coming on the basics, excuse me. I drink a lot of water. I'm sorry. So coming on, so I just thought, all these sequencing technologies actually rest on a principle which was so beautifully elucidated by Michael Moore. He published this paper on bulk segregant analysis, which by and large has been, most quoted paper, in the history of plant breeding. So this actually deals. So the current genotyping technologies also hinge around what Dr. Michael Moore actually postulated in the bulk segregant analysis theory, which he published in PNAs. So he actually gave an equation that you really don't need to have isogenic lines. Development of isogenic lines for trait discovery, I think, is a cumbersome process. And nobody has time and resources to actually generate those things. So this equation, which tells you that just by bulking, so if you see here on the right side, just by bulking, two extreme bulks, which constitute bulk 1 and bulk 2. Your bulk 1 is bulk of all the susceptible plants, and bulk 2 is bulk of all the resistant plants. And when you do genotyping on the basis of pattern, you can easily find out which particular marker is actually related to your allele of interest. And you see here the probability of an unlinked locus goes so low, 2 into 10 to the power of -9. If you just use 15 individuals, 15 resident individuals and 15 susceptible individuals. So this is the basis of all the current generation sequencing platforms. Like you have QTL-seq technologies like QTL-seq, RNA-seq, Indel-seq, bulk-seq, MuteMap, and things like that. So So I thought, I think it would be interesting for all of you to know a bit of this. So this particular slide actually tells you how it is actually wise to go for sequencing of pooled individuals rather than sequencing or genotyping of individual plants or progenies. For example, in an F2 population, typically you have population sizes ranging from maybe 100 to 1,000. So you would like to genotype all these, individuals. So this maybe is not required if you actually try and consider pool-seq, wherein you, as I showed in my previous slide, you bulk individuals together and you do sequencing. You gain efficiency manifold. Essentially, your sequencing cost is reduced to sequencing just two bulks, just two samples. So that reduces actually the cost, and also is pretty. So in this paper actually is from Schlorter et al. 2014 Nature Review Genetics. You can actually see this. It's a beautiful paper. So they demonstrate conclusively that once your pool size actually becomes more than 50, this strategy becomes really very, very attractive. So my sincere request is maybe all of you should also try giving a thought to all these pool-seq based strategies rather than just going for individual plant sequencing. Now, so I mentioned about MuteMap in my previous slide. So I just thought maybe I should also let you know about something called as MuteMap Plus, which was published by Fake et al. In 2013. So here, the beauty is you don't need, like in MuteMap, unlike in MuteMap, you don't need to actually cross your wild type plant with a mutant plant. Here, what you do is you just identify a mutant plant, heterozygous mutant plant in M2 generation, self-it. It actually segregates in a typical 3:1 ratio, and you do your SNP calling on the basis of differential SNP indices. You actually can very well tag your gene of interest, and this is a very, very attractive approach, which all of you should try at some stage of your career. Now, finally, on the general section, I also thought maybe I should let you all know about the JIVAs. JIVAs has been around on this side of the road for quite a long time, but then it has got its disadvantages, and the biggest disadvantage is its inability to handle anything which is having low electric frequency. So we have to actually with the change in sequencing technologies and the ability to handle data, I think we should be in position to model more than what actually is modeled currently, which is just additive model. So we need to go for G by G, gene by gene, and G by E environmental models. And also, non-additive models which actually lie untested, lie unutilized, I think should also be taken into consideration. And I think a lot of advances have actually been made in this direction. And we are indeed able to handle cases where minor allele frequency is less than 0.05. And this also involves handling cryptic population structures and family relatedness. As you know, association mapping, you need to really correct your population for structure and family relatedness. So all these things actually need to be taken into account before we actually take a step ahead in JIVAs, which actually is a very I would say attractive option to many plant breeders. Now, so finally we come to the crop in question. So the movie starts now. Pearl millet. So I'm sure some of you or many of you would have seen this crop, pearl millet. It's called as bajra, and this crop I would say is the crop which holds the hope of the humanity. because of its climate resilient traits. We, although I myself, I'm a rice eater and wheat eater, but I really appreciate this crop, not just because I work on that, because of its trait, which is friendly, not just to the environment, but also to the farmers and also to the consumers. So it is actually good on all the fronts. It's a crop which is climate change ready crop. I'm not sure if some of you would be able to believe that pearl millet seeds are able to germinate at soil temperatures as high as 65 degrees Celsius for 5 hours. I have one PhD student who is actually working on this, and we really pushed the boundaries. I mean, we had some previous reports from physiologists, primarily based at ICRISAT, who did some research work. So their limit was like 64 degrees Celsius. But now we have gone to 65, we have gone to even 68 degrees for 3 hours. And we find, in some of the accessions, we find variability. Seeds are able to withstand that kind of enormous heat, which I'm sure no other crop species would stand. So this is about seedling stage heat tolerance, which I'm sure Dr. Tara Satyavati would be talking in detail in her presentation tomorrow, because that is one of the biggest constraints in many areas, many pearl millet growing areas. So apart from seedling stage heat tolerance, pearl millet is also flowering stage heat tolerant crop. Crops like rice, wheat, and corn, they don't set seed once the air temperature goes on other side of 45 degrees Celsius, whereas pearl millet can set seed in air temperatures as high as 48 to 50 degrees Celsius. So that is the beauty of this crop. And plus, it doesn't demand you to dump, fertilizers, the kind of fertilizers you dump actually in rice or in corn. So this crop would be happy if you manage it with moderate management practices as well. And in areas like Rajasthan, where soils are shallow and infertile, it is the only crop. There is no choice; nothing else can grow. And that is why they say if you want to replace corn, you replace corn with sorghum, and if you want to replace sorghum, you replace sorghum with pearl millet. And if you want to replace pearl millet, then absolutely there is no choice. You cannot replace pearl millet, because that's the outer limit of your plasticity. So it also has got very high per day productivity. So we have found that it can actually produce biomass as high as 80 kgs per hectare per day. So it is comparable to or higher than switchgrass. So when we talk of capturing the sunlight and fixation of carbon dioxide, this is again crop of the choice. And another interesting stuff for this crop on the soil side is that it's a crop which actually can grow on a wide spectrum of soil pH. So in terms of acidity, this crop is as tolerant as finger millet. And in terms of sodicity or alkalinity, this is as tolerant as barley. And when you come to its nutritional traits, I think this crop is the crop of choice for people who are diabetic. Because of the presence of certain fraction of starch, which I'll be talking in my later slides. And it is also believed to be antihypertensive. It has got compounds which actually can dilate, can relax your blood vessels, and can reduce your blood pressure. And because of its high polyphenol content, it is also regarded as anti-mutagenic. It prevents against onset of cancer. And plus, it's the only crop which has got very high folic acid content. It has got 10 times more folic acid content than rice or corn. And you might know, especially India, things are okay. But if you go to West Africa, where food basket is very narrow and population is primarily dependent on pearl millet, so there folic acid becomes really critical. You might know some of the mothers here would know that in the first trimester of pregnancy and the last trimester of pregnancy, they would take folic acid tablets. So if you take pearl millet in certain quantity, you don't need to take folic acid tablets. So, and in some areas like Africa, you don't have access to these kind of supplements. And plus, it also, in US, it is used to produce something called as designer eggs. So the poultry fed on pearl millet would lay eggs which are very good in good cholesterol. In the sense, HDL fraction goes up and the LDL fraction comes down. And in terms of fodder, as I mentioned, it has got very high biomass content, and a lot of research is actually done on forage quality, and it has got no prussic acid problem or HCl problem. Excuse me. Now, resetting the clock back, pearl millet was always referred to as orphan crop, and we had, very, I would say, poor resources, genetic resources, and genomic resources until 5, 6, 7 years before. And you can see here these are some of the examples I have cited, wherein you see here the marker loci actually ranges from as low as 87 to maybe as high as 321, which is not great, comparing to rice or corn, wherein you have many thousand markers actually. So then we had this great milestone in the history of pearl millet, and we got the pearl millet genome sequenced, and not just pearl millet genome sequenced, we got many lines resequenced. So it was 1,000 genomes resequencing project, wherein we sequenced one reference line, which is TIFT23D2B1, which is called as well reference germplasm. So that was sequenced at 300x depth, and the rest of the lines were sequenced at varying, depths, which I'll be dealing later on. And all this was done under the able leadership of Dr. Rajiv Vashne, who earlier had sequenced pigeon pea and chickpea. And after this maybe was the turn of groundnut. So he actually helped in a big way to generate enormous genomic resources for this crop, which was regarded as an orphan crop till 2016. So this is about the 1,000 genome resequence project, which I was talking about. So in this, this is the reference line. It's the reference genome. It's the only reference genome available so far in pearl millet, and we are in the process of generating additional reference genomes in partnership with Corteva. So that will come very soon. But this reference genome actually is the one which is helping us out in doing SNP calls. And this was actually resequenced at 300x coverage. You can actually get all the statistics in the Nature Biotechnology paper. Apart from this, we resequenced something called as PMI gap. So this is actually a World Association Mapping Panel, which represents the genetic diversity of 27 different countries. And this was whole genome resequenced at about 1.8 x coverage. And in addition to this, we also sequenced actually parental lines of mapping population and B and R lines. So hybrid parent lines coming from different breeding programs. Ecosides breeding program and also partners breeding program were also sequenced at a slightly less coverage, which was about 1.6. And we also thought to study how the crop has evolved, how domestication has taken place. So we actually resequenced about 31 wild accessions as well, which actually helped us understand how exactly the crop behaved since its origin in West Africa. So this is something which I would like to share with you on distribution of SNPs and indels in pearl millet. So here you see, so pearl millet has got 7 chromosomes. So we had 7 pseudomolecules, and PG0 is kind of was the pseudomolecule which was assigned where not actually group. Other critiques actually. So if you can see here, actually the SNP, the number of SNP and the SNP density and the number of indel, it is actually enormous. And when you actually total all this, it comes over 49 million, which actually is enormous.
उसका नेट चला
Now, coming on the parental line side, you have more than 12 million SNPs. So I mean, with help of this, now the Pearl millet community is in a really good position to actually use this genomic resources for trait mapping. Now, coming to some genes which we thought were interesting in pearl millet, and so we studied the expansion and contraction of different genes in pearl millet vis-a-vis other species listed over here. And you can see here when we did the pathway analysis, we could find genes related to cutin, suberin, and wax biosynthesis pathway as expanded. There's a lot of noise in the background. Hello?
Excuse me.
There's a lot of echo coming in the background. Okay, yeah, it's better now. So we studied about 384 gene families, and we came to know that cutin, suberin, and wax biosynthesis gene families were considerably expanded in pearl millet, and they had a role in the amazing plasticity pearl millet has, in terms of drought and heat tolerance. So I'm sure some of these genes would also have counterparts in other crop species, and if they're identified, maybe they can be used to impart heat tolerance in those crops as well. This is a slide on the synteny between pearl millet. Just for the sake of reference, I just wanted to show you that pearl millet is actually closest to Setaria and also sorghum. And it has got reasonable degree of similarity to Oryza and Zea and Brachypodium. Now coming to so after this sequencing was done, and we had access to about 29 million SNPs on the station mapping panel, PMI gap, and similar number of SNPs on the BNR lines. So we actually started from about 32 million SNPs, and we tried to design a virtual SNP array. I mean, so this SNP array. So this is part of one of my postdocs who is an PDF fellow with me. So he is actually his name is Rambaran, and so he is doing this work, wherein he's trying to filter down those large number of SNPs to a reasonable number. So currently the number stands at about 68,000, but we'll cut it short and we'll try to make a kind of 60K virtual array. And all this information actually would be posted on ICRISAT's data repository, from where anyone can actually download and use these sequences in their studies. Now, I just thought I should also let you know in brief what are the traits which we have actually mapped in pearl millet. So you can see here, I mean, it's a big basket wherein we have mapped traits related to yield and yield contributing traits. On the left side, you see here the details. On the right side, we have mapped traits which relate to grain and forage quality. So which relates to traits which are important for nutrition from human perspective, and also which are important for the cattle, which relates to the forage quality. And you see we have traits which relate to biotic constraints, dunny mildew, blast, and rust. We have abiotic constraint. So you see here treadmill drought tolerance. You might know pearl millet. In pearl millet, we have had a long haul of almost 2-decade long study on terminal drought tolerance with our physiologist, and we have actually got hold of major QTLs which have been verified and validated in different genetic backgrounds. which actually take care of these component traits responsible for imparting terminal drug tolerance. And on other traits, we have, for the first time, we have actually produced something called as heterotic gene pools in pearl millet using molecular markers. And we have also got hold of, in pearl millet, I didn't tell you, in pearl millet, the major market is of single cross hybrids. So you need to generate, in the seed parents and the pollen parents, and you need to have a good estimate of the general combining ability. And that way, I think we have taken a first step wherein we have actually found out loci for general as well as specific combining ability. So this is a paper which actually deals with a plethora of traits. If some of you are interested, you can actually see it has got details of mapping traits which are related to phenology and which are also related to grain yield related traits. So I would not like to get into details of all this. Now, coming to, so we have at ICRISAT we have different kinds of mapping populations, and I would not like to get into details of that. So these are biparental mapping populations and your nested association mapping populations. We have reverse genetic populations in two genetic backgrounds. So we have produced these trilling populations. But I thought maybe this would be of some interest to you. So I was talking about this panel. This is called as the World Diversity Panel, and it represents genetic diversity of 27 different countries. And this panel has been used to map a lot of traits in pearl millet, which relate to grain quality and disease resistance. So blast has actually emerged as one of the major, diseases in pearl millet in last 4-5 years, and this panel actually is serving a good, base of having lines which have got very good levels of resistance to blast. So we are in process of mapping blast resistance, genes using this particular panel, and we have used this particular panel for mapping other different traits. So this is an example of GWAS we did using candidate gene approach. Actually, you can just, I would not like to get into details of this. So this is from +1 we published. You can actually, if some of you are interested, can see that paper. So I was mentioning about this combining ability paper. So this is what it is. And we have actually found some really good associations. And apart from that, we have also worked on downy mildew. So if you were to ask me, Rakesh, you let me know the biggest constraints of pearl millet. I would say number 1, number 2, and number 3 biggest constraints of pearl millet are downy mildew, and downy mildew. So downy mildew, of course, now blast is our additional headache, but downy mildew is one pathogen which actually has got enormous spatial and temporal variability. No hybrid stands in market for more than 3 to 4 years because of the enormous, pathogenic diversity it has got in farmers' field. So it defeats all the deployed resistant genes. So there is a constant race, with the pathogen, and also it has got a special, variability in the sense, any hybrid which is actually resistant here in Hyderabad can actually be susceptible in Delhi or in any other location. So we need to actually keep a tab of how exactly the pathogen dynamics is taking place and how we are actually deploying these R genes to control, this particular pathogen. And so far, actually, whatever QTLs we have mapped, they were all kind of small effect QTLs, 5%, 8%, 10%. But until recently, we, came across, I'm sorry. So we came across these major QTLs from some of the lines, which actually So I'm not sure if this is very clear. So these peaks are against three different epathotype isolates. These are three virulent isolates from Rajasthan, Gujarat, and Haryana. And you can see here, they are actually co-localized. And they are also major effect QTLs. So this is a good news actually for the community. By deploying these kind of QTLs, we I think should be able to have strong levels of resistance. But then again, how long these QTLs will stay in farmers' field remains to be seen. So I thought maybe I should also speak about one of the exercises we are doing here at ICRISAT, which includes SSR to SNP conversion. You might know in the past, a lot of SSRs have been used, and now because of the change in the platform, I think many of these are becoming redundant. So we actually are trying to convert these SSRs to SNPs. And so the strategy which we follow here is we actually do amplicon sequencing, and we get the amplicon sequenced. And obviously, those amplicons would have a lot of SSR motifs. So we actually walk on 3' and 5' region of that particular region, of course, aligned against the reference genome. And then we call the SNPs, and we design Caspase, and then verify and validate them. And we are able to get, in some cases, really, clearly differentiated, signals between two parental types here in red and blue, and the heterozygotes. Now, again, coming to downy mildew, which I was mentioning. So the challenges actually are manifold. Not just in terms of pathogen variability, which I was just talking about. There is a challenge on part of crop as well. The crop is highly cross-pollinated, and it suffers from severe inbreeding depression, which means some of the lines, if you start selfing, post F4 or F5 generation, you will not get any seed set. So you need to resort to sieving, and also some of the lines are photothermosensitive. And plus, most of the mapping populations actually have segregation distortion, which Amita was talking about. So segregation distortion is a major bottleneck in mapping traits of interest in this particular crop. So we have actually, to get over this, we have also generated chromosome substitution lines. And if any one of you is interested, they can actually go and see this. This paper is available, I think, in Crop Science. So we have some donor parents. So I just wanted to show you how exactly downy mildew resistance mapping looks like. So this is example of linkage group 1, but like this, you have example from other linkage groups as well. And you can see here, these are all small effect QTLs, and these all govern race specific or location specific disease reaction. So you can see here, so on the right side, these are all different donor parents which we have used to map, and these are all color coded. And on the left side, you have different locations coming from India and Africa, and there's a complex interplay between the donor lines and the way they behave as far as different pathotype isolates are concerned. So it's a complicated pathogen. But right now, in pearl millet, we have achieved a reasonable degree of comfort in mapping, in deploying our resistant genes, but we are quite shaky on BLAST, which I would mention later. So again, coming back to Dr. S.K. Rao's point, so this is all about science, about publication. So what it means actually to a farmer. So here you all will be happy to know that the first molecular breeding project in any crop in India was released in pearl millet. It was not in rice or corn. So this improved version called as HHB67 improved was improved for downy mildew resistance. The original version was which is called as HHB67, which Dr. Bhardwaj also mentioned. So that original hybrid went susceptible to downy mildew, and then we introgressed two major downy mildew resistance QTLs and put it in the male parent coming from CCS HAU. So this particular hybrid actually is released from CCS HAU as HHB67 improved. And you would be happy to know that out of about 8.5 million hectare area of pearl millet, this is cultivated in more than 10% cropped area, which is about 870,000 hectare. And it actually so this entire activity was part of a BBS-RC funded project, and we did some impact assessment study at ICRISAT, and we found that in the year 2011 when the study was done, we found that in one season the benefit to the farming community was worth US 13.5 million. So it's a huge contribution. It's much more than what the project actually was. The total outlay of the project was. So that project was almost under $2 million US dollars. And you see the returns on the investment. You get more than 13.5 million every year, and not just from the crop but also from seed production. So seed production of HHB67 happens mostly in Andhra Pradesh and Gujarat. And the seed producers get a lot of income from production of the seeds of the parental lines and also hybrids. So this is something on the data of HHB67 and HHB67 improved. So the target rate was downy mildew, you can see here. So on the original HHB67, which is coded in red, you see here downy mildew incidence was almost close to 100%, whereas in HHB67 improved, which had two major QTLs for downy mildew resistance, the incidence was less than 5%. So and it also had superiority in other traits. For example, the grain yield actually increased from 1.8 to 2 tons, and also the forage yield. So many times the linkage drag is not always negative; it could be positive as well. So this is another slide wherein we actually mapped downy mildew resistance QTLs. So as I was mentioning, downy mildew pathogen is extremely variable, and we just wanted to study the tricks of the pathogen as to what actually makes this pathogen so smart. So we got this pathogen sequenced with help of our partners, including Dr. Chandra Nayak and Dr. Tara Satyavati. So we sequenced this pathogen, and surprisingly, the genome size was actually on higher side. We had tough time convincing the reviewers that this Sclerospora pathogen doesn't have genome size typically expected between 40 to 50 MB, but then this one is close to 300 MB. And you can see here the number of genes. The number of genes compared to its genome size is astonishingly high. It's over 65,000. So this also indicates that maybe this is the reason why this particular pathogen has got so much of plasticity and intelligence to defeat all the deployed, resistant genes which are put across. Now, coming to BLAST. So as I was mentioning, BLAST is the new pathogen which we maybe has come from rice. So it's a cousin. Pyracularia gracia is the causal organism of foliar blast, and it is blasting us all around. I tell you, it's an epidemic-like situation. Dr. Tara, when she presents tomorrow, she'll maybe explain in greater details as to how this is causing havoc. If you go to some fields during rainy season, you can actually smell the stink which comes out of rotting of the leaves because of this pathogen. And we have so far very less number of donor lines, resident donor lines, which are pan-India. So these all govern resistance against particular pathotype isolates coming from a particular region. So in this aspect, it is where PMI gap actually has come to our rescue. And this is part of PhD work of student Dr. Mukesh Sankar from IARI. So he actually has evaluated this particular panel, PMA gap, in different locations, hotspot locations of the country, and also in greenhouse trees. And we have got some very encouraging and exciting results. So this is one example. They say seeing is believing. So on the left, you can see here, this is one important hybrid called as HHB 146 on the left, and on the right, it's HHB 146 improved. So what we did is we moved one of the blast resistance QTLs, which was located on linkage group 4, to the seed parent of this particular hybrid, HHB 146. And then cross that with the original pollen parent, and this is what we got. You see here. So here on 1 to 9 disease scale, if 1 is highly susceptible and 9 is highly resistant, although many people follow it in different way, but going by this scale, maybe this would be highly susceptible, and this would be highly resistant. Maybe the score will go somewhere close to 9, and here on the left, the score will be somewhere close to maybe 2. So you can see here, blighted growth, coalescing of these islands, and also there is an attack on the midrib, and you see here one of the culms is also broken, because it rots. Okay, so now coming to rust. Rust is also important in pearl millet, but not so much like blast or downy mildew. But then it is important, and it comes late in the season, so it is important more for maybe forages. So here, with help of one PhD student, this is one PhD student's work. Her name is Supriya, and she is right now working with Dr. Tara Satyavati at Jodhpur. So we found actually a major effect QTL on linkage group 1, and this has actually been validated. And this is an attractive target actually to be used for molecular breeding. And this is a major effect QTL. It actually governs 69% phenotypic variance. Now, coming to another important work. So this is part of a collaborative project between University of Cambridge and ICRISAT. POE is also involved. NIPGR is involved. So they are working on different crops. For example, POE is working on wheat, and NIPGR is working on foxtail, and ICRISAT is working on sorghum and pearl millet. So here we are trying to actually look into how exactly pearl millet behaves as far as nitrogen use efficiency is concerned. Because the soils where pearl millet is grown are extremely depleted soils in terms of nitrogen and phosphorus. So of course, we have kept phosphorus away from this project. So this project is only about finding out genes related to nitrogen use efficiency. And we have got some excellent results, actually. So we have got hold of some good lines again from this World Diversity Panel, PMA gap, which we actually evaluated for 3 seasons in 3 different nitrogen conditions. So 0%, 50%, and 100% of their counter doses on depleted fields. And we got really exciting results. And we are also actually working on some of the candidate genes related to allele aminotransferases, which actually is thought to play a major role in nitrogen use efficiency, and glutamine synthetase and glutamate synthetase. So this work is in progress, and very soon I think we should be in position to communicate this to some of the peer reviewed journals. So now coming to one of the important traits for pearl millet I was talking about. So pearl millet, as you might know, is branded or rebranded as a NutriCrop for the obvious reasons. And here we have actually, as Dr. Malu was mentioning, and Dr. Bhardwaj was also mentioning, we have tried to target 2 important micronutrients, iron and zinc. So, excuse me. So So in pearl millet, the typical variability for grain iron is from say 25 to about 135 ppm, and zinc again about 25 to about 110 ppm. Whereas if you compare this with other crops like rice or wheat, the iron and zinc levels are the base levels are low. I mean, it could be as low as maybe 10%, 15%, or 10 ppm or 15 ppm or 20 ppm. So in pearl millet, we have ICRISAT has also released in association with partners, various biofortified cultivars wherein the iron levels are on other side of 80 ppm. So we basically are on the discovery side. So we have worked in association with national partners, including Dr. Tara Satyavati, the QTLs responsible for high grain iron and zinc content. So this was the first paper we published. So this is work of the PhD student of Dr. Tara Satyavati, and we found some very good signatures of favorable alleles spread across different pseudomolecules. So we found that so this was all SSR centric work, and we found that one of the SSR markers, which is XIPS 0810, overlapping with aspartic proteinase gene on pseudomolecule 3. So this is interesting, and this needs further investigation. Now coming to other work, we have also published a major QTL paper on major QTL, which actually gives, you can see here, the R square is on the other side of 50% for zinc, and it is close to 20% for iron, and these are all co-localized QTLs. So that is very interesting thing in pearl millet we have found from all these biparental mapping populations. So that means if you breed for iron, you also breed for zinc and vice versa. And this could be related to these ionic channels which are found in these crops, which are non-specific. So a channel or the transporter which actually picks up iron also picks zinc and vice versa. So this could be the reason. So we also had a different mapping population. So this is AIMP 92901 was a donor parent which had iron content on other side of 110 ppm. So there also we found on a different linkage group co-localized QTLs for iron and zinc content. So these are all very interesting, clues. And if you want details on this, you can just download our paper in Genes. It's an open access journal. So this is in detail about different QTLs which we had, identified across environments and locations. We also did some differential gene expression study. This is work of one of my PhD students, Mahesh. So he did this work, and we found clear-cut, family of genes which actually were expressed differentially between high iron line and low iron line. So in this case, we chose the two mapping population parents, and of course, we had check as well. And the interesting thing is we could find a crosstalk between differentially expressed genes, its microRNA apparatus, and also its overlap with the QTL interval. So we actually, we dissected the QTL, and we wanted to see what exactly lies in the QTL. So some of the signatures were common. So that's very interesting, and we have actually submitted this paper. So it has come back to us. We submitted this paper in Scientific Reports. It is back for the rebuttal. So let's see how that goes. Now, so this is a major milestone in pearl millet, I would say, crop improvement. This was work of again one of my PhD students, Radhika Ramya. So she did this work, and for the first time we could identify heterotic gene pools in pearl millet. So as I was mentioning, pearl millet, there is a lot of market presence for hybrids. So unless you breed for hybrids, you're not in market, So you got to breed hybrid parent lines, which are B lines and R lines. So here, if you see, all these lines which are mentioned in blue line are your R lines, And all these lines which are mentioned in red, I'm sorry. So all these lines which are mentioned in blue are B lines, and all the lines which are mentioned in red are R lines. So you can see here, I mean, so we did SSR genotyping. So we just wanted to see how exactly some of these SSR markers, especially coming out from GCP project, actually behave as far as allelic differentiation of all these lines were concerned. So we started with our repository of about 750 SSR markers, and then came down to maybe 100, and then did this analysis, and we found clear-cut heterotic groups on the basis of this genetic differentiation. And in this process, we actually generated data from 4 different environments. And this PhD student actually, on a lighter note, I mean, that time she was was getting married. So this PhD student, her would-be in-laws had seen her, maybe 6 months or 8 months ago. And by the time she finished experiments, they saw her again, and they got confused whether it is the same girl or some other girl, because she really spent a lot of time under hot sun and got her skin burnt. So what I mean to say here is, I mean, if you really have all these genetic and genomic resources in place and make good use of them, you can really dig out some interesting piece of information. Now, on a general scale, on GS, my previous speakers have talked a lot about this, especially Dr. Bharadwaj. So the genomic selection in Pearl millet here is slightly different because of the fact that you are not, carrying forward the selected line as a variety. Okay, so you build your model training cycle individually for the 2 parental lines, which are B lines and also R lines. So in this, apart from in Pearl millet, per se performance is important, especially for seed parents. You need to have good seed set on the seed parents, but that's not all. We need to actually move ahead from per se performance to combining ability studies. So in this genomic selection model, we also try to study the general combining ability. And once we have general combining ability estimates on both B line side as well as R line side, so we then also get over to the next stage and try to estimate the specific combining ability on the basis of information we generate from selected testers. So it's slightly complicated, genomic selection in Pearl millet. And for any other crop where you have hybrid parent lines is slightly complicated, because you also need to take into account GCA as well as STA, SE estimates. So this is another slide which actually talks about whole genome prediction model, which we actually did in our Nature Biotechnology paper. So here we got reasonably good prediction accuracies for many traits. And we had used historical data actually coming from Pearl millet hybrids and the parental lines. And this is the latest which we are actually trying to do on genomic selection. So in this we have actually gone a step ahead, and we are trying to see how different kind of SNPs, whether genic or intergenic, or those lying in the promoter regions, or those lying in the exonic regions, like that, are able to, contribute to prediction accuracies. And we have got some interesting, results, because not all SNPs coming from different genomic regions give you, equal type of prediction accuracies. Now, coming to last few slides. So Dr. Malu was mentioning that let your food be thy medicine, so in that direction, we are, using Pearl millet as a vehicle to tackle diabetes. And you might know diabetes is a global problem, especially for India, wherein every 3rd individual is either diabetic or pre-diabetic. So we actually are making use of the starch fraction which is present in Pearl millet. So we had a couple of GCRF projects from UK, wherein we studied variability of the starch fraction in Pearl millet. And we found that pearl millet is really very high in a portion of starch called as SDS, which is slowly digestible starch, and also another fraction of starch which is called as resistant starch, or RS. So we then on the basis of extensive phenotyping, we identified some of these lines which were really very high on SDS and RS, because these two traits can actually bring your glycemic index by appreciable quantities. So we used these lines. So we evaluated the parental lines, and we also evaluated the test cross hybrids. So this picture which you see on the left is from Bowel, and he is our breeder from Bowel. So you can see here, and it actually, this location falls in A1 zone. So A1 zone in pearl millet is regarded as the most dry zone, where the rainfall is less than 350 mm. And the crop, you can see here, even in this stress condition, it was really very good. And most important thing was, it was also resistant to blast and downy mildew. So we are trying to take this hybrid forward, because just like iron and zinc, this trait also is a silent trait. So on one side, you understand the importance of this trait, but on other side, you would also like to make this hybrid or whatever product you develop, agronomically superior, because otherwise then farmers will not be able to actually grow these kind of cultivars. So we also did some association mapping, and we got hold of some important signals on chromosome 3 for SDS, which is slowly digestible starch. But then this needs to be really validated. Now coming to forward breeding. So all this was on discovery side. So this actually feeds into forward breeding pipeline we have here at ICRISAT, wherein we use all these validated markers to downstream breeding pipelines where they're used in breeding program. So we have a suite of 10 SNPs, which we actually developed from DDR sequencing by fine mapping the linkage group through drought tolerance QTL. Of course, I didn't mention much about that particular QTL. So that is a validated QTL against different genetic backgrounds, and we also know the component traits for which this particular QTL is responsible for. So we targeted that QTL, and we fine mapped, and we got hold of about a suite of 10 SNPs which are used in forward breeding. And Dr. Tara Satyavati is getting a lot of lines coming from the A1 zone, the dry zone, which she will be talking about in detail tomorrow, genotyped with these markers. And the other stuff is iron and zinc. So we have actually targeted the linkage group 3, iron and zinc density QTL, and we have got hold of some SNPs. We are also developing SNPs for other QTLs coming from other donor parents. So it's all in pipeline. Now, lastly, so this is the latest development here. So here at ICRISAT, we are trying to develop something called as a QC panel, quality control panel, which is SNP based. So this panel will be able to differentiate more than 99% of the germplasm and breeding lines, which come from global breeding programs. So we have taken into account lines coming from Africa and also lines coming from India. And Dr. Tara has provided a big support actually in this direction. So we again made use of the 1,000 Genome Project data, and we actually partnered with Corteva Agri-Sciences. So they provided some SNPs using the same data, and we had our pipeline of SNPs. We verified those SNPs on a small panel of lines. And right now we have about 30 SNPs which actually have gone for final verification and validation stage. So once we get information out of that, maybe we'll cut that number around 20 and try to make that as a global QC panel, which can be used not just in breeding programs, but also in seed systems, because you need to keep a track of purity of your cultivars, of the seed produced at different levels in the seed chain. So this is something which actually would come very handy as far as seed systems is concerned, and it is one of the weakest links actually in pearl millet improvement. So finally, acknowledgements. So all this work obviously cannot be done in isolation. So ICRISAT is partnering in a big way with EQUIP, All India Coordinated Pearl Millet Improvement Project, currently led by Dr. Tara Satyavati. And we have other ICR ICRISAT programs which actually help us align our discovery strategies with those related to the requirement in the market. We had support from Department of Biotechnology and also Department of Science and Technology, International Livestock Research Institute, ACAR, then DFID, BBSRC, and lastly, Bill and Melinda Gates Foundation. So, thank you very much. So at ICRISAT, we believe in teamwork, and my previous speakers also, talked about this because, nowadays this science cannot be seen in isolation. You have to work with different teams because you're not expected to be master of all, but at the same time you cannot be jack of all. So you are expected to take care of your domain of expertise and liaise with your teams, and then make sure that it actually flows seamlessly in a product development pipeline. Thank you very much for your attention.
There are some questions from the.
Let me stop sharing my screen presentation. Just a minute. Yes, please. So pests such as stem borer is a problem. Any genetic solution for the pest? Yeah, this is a good question because all these years, pearl millet was supposed to be a crop which was reasonably resistant to many pest crops, including stem borers. But now we have seen that it is becoming susceptible to some of the stem borers and also to head miners and root grubs, and of course fall armyworm. So, actually, so far we have not, attempted any concrete efforts in this direction. But I think we have a reason to target traits such as not sure stem borer because it is actually sporadic and comes in certain locations. But of course, root grub is a big problem in some of the areas in Rajasthan. And also, maybe if the spectrum of fall armyworm, expands, we may have to look into pearl millet as well. How exactly the repository of genes are working against these pests. So I think just change. Somebody asked about rancidity. Yeah, so rancidity is one trait which is extremely important in pearl millet. Good that you asked that question. And in fact, thanks to Dr. Tara Satyavati again, because we at ICRISAT were harping about this particular trait because it is regarded as its heel. It has got all the good stuff which I talked about, but at the same time the flour, the atta actually turns rancid within hours to days after milling because of so many factors, which includes fat as well as the enzymatic content. So actually, we are working in this direction. We have already started to investigate what are the genes, the pathways which are actually responsible for rancidity. And it's not very simple. I mean, it's like if you just select lines which are low on fat, you expect these lines to be low on rancidity, but that's not the case. So that shows there are many factors actually in the background which are playing their part. And we have made a beginning in this direction with Dr. Shelly Praveen. So she was brought into our fold with help of Dr. Tara Satyavati. So we are trying to take a look as to what is happening in pearl millet. And so far, I would tell you, we just have some candidate enzymes. So we know enzymes like polyphenol oxidase or peroxidase. They play an important role in rancidity. But at the same time, they also have role in initial plant vigor and growth. And also they impart resistance to stored pests. So we have to actually do a balancing act as to how we address rancidity, but at the same time don't take away the stuff which is actually needed by the crop. Yes. So the next question is, what is the role of microbiome research in cereal yield enhancement and quality? Okay, yeah. So this is a very good question, but slightly outside my area of expertise, because I'm basically a plant geneticist. But then, yes, your question is valid, because nowadays, I mean, we shouldn't just be talking about the crop. We should go a couple of paces ahead. So we should talk about the crop. We should be talking about the food which is made from that crop, and then we should also be talking about how that crop is actually absorbed in human gut. What is the role of gut microflora and gut epithelium chemistry? So we at ICRISAT, we have a group which actually is looking into this particular direction, and we have actually selected some lines which are low on are high on iron and zinc to study these kind of things. So as for now, I'll not be able to comment anything, but yes, I mean, we are working on this, and we are trying to see the role of gut microflora on these various parameters. Yeah, so there is another question: why consumption of pearl millet is much less as compared to rice, wheat in India? Yeah, this is a good, very interesting question. So it's a very complicated question as well. I mean, it is related to so many factors. So one is the way our habits have changed over a period of time. If you see how our forefathers used to eat. Maybe you can check with your grandfather. I'm sure he would tell you that they had a lot of millets on their platter, and these over a period of time, and maybe this is also one of the negative sides of green revolution, which actually pushed out many of these important cereals out of food basket. But now I think government is realizing, and many of these millets are actually on a comeback spree. And on individual level, I think you should also realize the nutritional benefits which these millets actually provide you. So once you have this awareness, especially in the middle class, I think it can go a long way in pulling the demand up. So once demand goes up, then farmers will be tempted. Farmers would find attractive actually to grow this crop, and maybe they'll get better price in the market. So, whether high chromium in pearl millet is desirable character for food purpose? Okay, yeah. So, so anything, it is Lord Buddha's, as per Lord Buddha's teaching, one should always fall in the path of medium, or mean. So madhyam marg, they say. So any element, if it goes beyond a particular limit, it could have many undesirable effects. And in this case, chromium I was pointing out, because if you see the chromium content of other cereals, it is on very low side, whereas in pearl millet, chromium content is very high. And this chromium content is at the same time not toxic to your body. In the sense, it's not so high that it starts hurting you. But on the contrary, it actually helps you get balanced diet. And it plays a role actually in diabetes. Chromium has a big role in diabetes reaction. So if you're running low on chromium, you can actually become pre-diabetic or diabetic, and you can even have complications related to hypertension. So all those things are there. It needs to be investigated. Yes. Another question: can pool seek work to develop barcode for identification of different species? Yeah, this is a fantastic question. Yes, yeah, I think that's a possibility, but that depends how you are actually modulating your strategy. Okay, so if you see the GBS pipeline, in GBS pipeline, many things are barcoded. Okay, so your DNA signature actually goes with particular barcoding. So if we apply similar kind of stuff over here, yes, I mean, it can be done here as well. So there is a question: despite impressive nutritional qualities, why millets has not come out in mainstream crop? Well, pearl millet is only recently maize has overtaken pearl millet. Otherwise, if you look in terms of area, it was rice, wheat, and then it was pearl millet. Now pearl millet has been pushed to fourth place, but still that is not bad. Yes. So I mean, there are, as I was mentioning earlier, there are various factors actually related to market dynamics and demand. So once we take care of those things, I think millets can do very well. If you look into the footprints of millets, compared to rice or energy footprints, it is very low. I mean, see, when you eat a kg of rice, you see how much energy has gone into produce 1 kg of rice, how much water has gone into production of 1 kg rice, vis-a-vis, pearl millet grain. So that way, environmentally, I mean, you are on a sound side, and all these things really actually need to be put before the stakeholders and the farmers. Yes, Dr. Bhardwaj, you're raising your hand.
Yeah, Ashok, I think we forgot our roots. Earlier, it was only millets that was being consumed in our
Yeah,
modernization hurry. We lost
yeah,
them, isn't it?
that's what I told. If you were to ask your grandfather, he will tell what exactly they were eating then. Yeah. So another question: which omics technique can offer much needed climate resilient drought and heat stress resistant high yielding millet crops? Mostly they are facing drought and temperature stress in their seasons. So it's like it depends. I mean, there is no I would say a blanket recommendation on this. So it actually depends at what level you are, what are your project goals, and what exactly you would like to achieve. But assuming that you have access to required funding, I think you should go to all these next generation sequencing based technologies which I talked about in getting to the root of the matter, including drought and heat stress. Of course, I mean, phenotyping, as Dr. Bhardwaj was mentioning, is not an easy job when it comes to both drought as well as temperature stress. In drought, we have been able to take care of just terminal part, but then if you go to the typical agroecologies in Rajasthan, Gujarat, and Haryana, where drought happens on a very frequent note, you would see it is not just terminal drought. I mean, you have various kinds of drought, and it's an interplay of complex factors. So yes, I mean, precision phenotyping in the right context, I think, would help us go there. So there is a question: how do pearl millet PDS system in rural and urban areas of our country? Oh my God, this question is something which I cannot handle. But yes, I can tell you that recently government has actually hiked the procurement prices. That's a good news to pearl millet community. And I think government is taking care of distributing pearl millet in PDS system, because government appreciates the importance of pearl millet in nutrition, human nutrition. But how it goes, I have absolutely no idea. I'm sorry.
Thank you very much, sir. We have covered all the lectures today in times. I am thankful to Dr. Nagendra Kumar Singh, Dr. Sri Bhardwaj, Dr. S. Amita, Charu Rama Mitra, Dr. S.R. Malu, and Dr. Rakesh K. Srivastav. Today, about 582 delegates were participated in our session, and they have raised good questions, and I appreciated the most of the informative information in your all talks. I am once again thankful to you. I am thankful to Dr. Rao, Vice Chancellor of this university, to give a chance, facilitated to us to have a such conduction at our university. Thank you very much, sir.
Thank you, Rakesh.
Thank you very much. Thank you very much for organizing this wonderful this webinar. Thank you. Namaskar. Stay safe.
Thank you, sir.
Thank you.
So we will be back tomorrow at 10 a.m. Technical session has been shared with you. Thank you.